Hello everyone, a very warm welcome to Ashish Biology. I am Ashish Kumar Das, student of M.Sc Animal Biology, University of Hyderabad. So in this video, we'll discuss about mark and recapture technique, and this technique is from the unit of population ecology. This video will be useful for BSc and MSc students, CSIR NET aspirants, DBT JRF aspirants, ICMR JRF aspirants, ICAR NET aspirant and get life science aspirants. And at the end of the video, we will discuss some CSIR NET question related to the topic. So if you are new to this channel, please subscribe the channel and press the bell icon for getting more updates. Now, first of all, see what we will learn today. We will see what is population ecology, that is, we will define population ecology at our level. We will see how population density is affected, that is what are the factors associated with the population density. Then we will see what is mark recapture technique, and you will also see how this technique is useful in counting birds and animals population, that is what are the special significance of this technique we will see, and many more. So first of all, before knowing regarding population ecology or population density, we must have brief idea about population. So what is population? That is, we use the word population randomly, but here we have to define it ecologically, so in a scientific manner. So first of all, this is a collection of individual of the same species that live together in an area. That is, the individual must be member of a same species and they must be living together in an area. So here you can see this is a deer population. All of them are the same same species, and they are living together in an area. Here you can see worm. These are also the member of same species. Now you can see the tiger population. All of them are the member of same species, and they are living together in an area. That's why they also constitute a population. And here you can see beehive. This is an excellent example of population. That is, here you can see. So you can see the beehive. All of them are the member of same species, and they are living together in a same area. That's why this is also a population. Now population ecology. So first of all, population ecology, what is the meaning of the term population ecology? So study of population, especially the population abundance. So population ecology is the branch of science that deals with study of population, and mainly among population it deals with population abundance and how it changes with time. That is, how the population abundance is changing with time. And it also deals with the pattern in abundance. That is, the abundance pattern means some places may be highly populated or some places may be less populated. So these are the patterns in abundance, and also the distribution of organism, how the organism are distributed in that particular habitat, and the mechanism. That is, for example, we human being are more attracted towards the habitat that is having more employment opportunity, better facility in terms of health care, education, etc. Similarly, your wild animals will be better attracted toward the places that is having better food resources and better prey, that is, more amount of resources they will like. So what are the attributes of population ecology? So first of all, what are attributes? That is, these are the characteristics or the aspects of the population ecology that help us to compare and measure population. So these are the aspects or characteristics that help us, that is, by virtue of which we can compare and measure population. So the example are population density, natality, mortality, distribution, etc. So we will discuss this. So first of all, population density. So what is population density? The most fundamental property. This is the most fundamental property to represent the size of population. So for defining the population density, we must know the size of the population. Then this is the important fundamental property. So in most of the cases, a population is represented by the population density, and this parameter helps us in many areas. Then it can be defined as the total number of population per unit area. So this represents the number of people or the number of organism or the number of individuals that are residing in a unit area. So this is represented by the formula or symbol n, and the formula is like this: population density equal to number of people or number of individual divided by land area. So ultimately we can define this as the number of individual per unit area. Now, what are the two types of the density? So, crude density and specific or ecological density. So, here what happens? Crude density means the density per unit of total space. So, here it would be SPAC. By mistake, I have written this wrongly. And the density per unit area of habitat space would be the fraction of area that is totally occupied by the organism. And this will be ecological density. So, crude density means the density per unit of total space. For example, there is a forest, it is having area of 10 square kilometer, and in that there is a mountain that is having 3, or consider there is a small lake that is with area of 3 square kilometer. So where you are calculating the crude density, you have to take the unit of total space, that is 10 square kilometer. And when you are calculating the specific ecological density for a tiger, for example, we can take the lake as the habitat for a tiger, because a tiger will not reside there. So here what we will do, we will calculate the ecological density, and we will take the actual colonized area as the 7 square kilometer, that is 10 square kilometer minus 7 square kilometer. So ecological density is always higher than crude density, because you are increasing the denominator, that's why the value will always increase, and the ecological density will be higher than the crude density. So determining population density by mark recapture method. So how will determine? So it becomes important when the number of organism is very high and this is not possible to catch them. For example, if you have to calculate the number of tiger in a forest, you can't catch them, and you can count them individually. So here the mark and recapture technique become important, and it is also useful. Here two examples are there. First of all, to count the number of birds visiting a lake for drinking water. Suppose beside your house a lake is there and birds are coming there for drinking water, and this is not possible for you to count them individually and know their number. So you have to use mark and recapture method. And to count the number of deer residing in a forest. So similarly, in a forest you can't go to there, you can't go there and you can calculate the number individually. That's why the mark and recapture become important. And the one important thing here, it will never give you result with 100% accuracy. So the result you are getting will not be 100% accurate, but it will give you an estimation, that is you can get a scientific estimation, or you can say that this is the approximate number of the organism present over there. So here this will not give you the data with 100% accuracy, but sure it will give you a scientific estimation. Now procedure, what is the procedure? You will see a small random population of the, that is random sample of population is captured and they are marked. So a small random population means you will not that is not specific that you will have to catch the organism from that particular area of the forest. You can obtain them, that is you can catch them from any area, and you will mark them with small tag, for example like rubber band. So marking here, it doesn't mean that you have to mark them with your pen or pencil. So here you will mark with small tag, or simply you will put a rubber band on their ear. Then the marked individual are released to disperse within the population. Then you will leave them, and after them they will allowed to freely mix. Obviously they, if they are that is dispersed in the population, they will freely mix with the whole population, and the population is resampled again. So again you have to resample the population, and the number of the unmarked and marked individual you have to record. So here the formula is there. So population size is equal to n1 into n2 divided by n3. So n1 multiplied by n2 divided by n3. So what are n1 into n3 you will see. So the number of individual caught and marked initially is your n1. That is the first sample that you have obtained, that you have caught and marked them with a tag or any rubber band. Then n2 is the number of individual caught in the second sample, that is the second sample you are collecting. And n3 is the number of marked individual recaptured, that is the number of marked individual recaptured in the second sample. So here the formula become n1 into n2 by n3. This become more clear when we will discuss some problem on this. So this is most suitable for animals that move around and are difficult to find. So this is important when the animals move around and they are difficult to find. So other name of the technique, sometime in examination question come and they use the other term, they will not use mark recapture, they will use other terms. So what are the other terms? The other terms are capture recapture technique, capture mark recapture techniques, site resides, mark release recapture, and mark recapture. So you have to remember names of all these techniques so that you will not be confused when you are facing a question in the examination. So now we will solve some question related to this topic. So this question is from the CSIR NET 2017 June. So in an experiment to determine the number of rats in the field, 80 rats are initially captured, marked and released. After 1 month, 100 rats are captured in the same field, of which 20 were previously marked 1. So based on the above observation, estimated population of the rats in the field will be. So this is direct question. So first of all, we have to see what is N1, N2 and N3. So here, as the question is suggesting, 80 rats are initially captured. So this will be N1. So N1 will be equal to 80. So after that, what happens? Again 100 rats are captured in the same field. So that is recapture. So recaptured means it will be N2. So multiplied by 100. And after that, what happens? 20 of them are marked. So what are the 20? They will be N3. So it will be 20. So here you can see, we will divide this 20 of 80, we will cut this. So it will be 400. Answer will be 3. Now you can see another question. This question is from CSIR NET 2018 December. So in order to estimate the population size of a fish species in a lake, the researcher captures 100 fish from the lake and mark them with the colored text. So here N1 equal to 100. Then a week later, the researcher returns to the lake and catches 150 of the same species. So N2 will be 150. Then what happens? And among them, that is the 150 captured, we are having 25 of them are previously tagged 1. So N3 will be 25. So that is assuming no migration or emigration occurred, that is no no fish have migrated from the lake to other lake, or no other fish have come to that lake from other lake. So calculate the number of the, that is the fish species. So here the formula will apply. So what will be the formula? So N1 into N2, that is 100 into 150 divided by 25. So N1 into N2 divided by N3. So here it will be 6. So the number of, that is the number of the fishes will be 600, option 3. Now this question is from CSIR NET 2013. So a grasshopper population is being assessed by the capture, mark, release, recapture method. So here you can see the name is changed here. Usually mark recapture is used, so there is another name, but you don't have to confuse yourself. So on the first day, 100 grasshopper were captured from a given area in 1 hour time, marked and released. On the next day during recapture, 10 marked and 90 unmarked grasshopper could be found in the same time period from the same area. What is the estimated population? So here you can see 100 grasshopper are captured from the given area in 1 hour time. So N1 would be 100. Then what happens? 10 marked and 90 unmarked grasshopper could be found in the same time. So what will be N2? N2 will be both marked and unmarked. So N2 also will be 90 plus 10, that will be 100. So N1 into N2, we are getting 100 into 100. And the N3 will be, that is the marked sample obtained in the second sample, that is the marked individual obtained in the second sample. So it will be 10. So here it will be 10. So where we are multiplying this, we will divide, and the answer will be 1000, the third option. So in this way you can solve the question, and this topic is very easy, and you have to remember only one formula, so scoring become easier here. This is the end of the video. Thank you for watching, and if you are new to this channel, please subscribe the channel and press the bell icon for getting more updates.